Hello, everyone. My name is Adrian Roth, and it's a great pleasure to be here and to be able to talk to you online on this webinar. I work for Roche pharmaceutical company based in Basel, Switzerland in clinical development, and I've worked for quite a few years in preclinical safety assessment. And for the next couple of minutes, I would like to talk to you and explain to you what's our interest in organ-on-chip, how we use them, where we see application during drug development, and what might need to be done to fill the gaps that we are facing and also to address regulatory requirements. So first of all, what is driving our interest? We would like to renew the classic drug development paradigm. I'm sure most of you are aware of TAR, where we start with identifying new targets and selecting hits. This is typically done using mouse models. You have a disease model that recapitulates a certain disease. You then start screening on your target using cell lines. Once you have identified a valid candidate, preclinical development start, which is typically very animal heavy, using different types of animals, a rodent and a non-rodent species, to enable them for your final candidate to enter clinical development. Now that has worked pretty okay for many, many years, right? Though it's not perfect. So this is, there's a number of publications out there. This is one of the more recent one from a colleague, that has been published. Why do certain candidates fail later on in clinic? And as you can see here on this graph, it's particularly those adverse events that lead to clinical attrition. Particularly those are not really well predicted, of course. So this is where we definitely would like to improve. So we would like to have systems in place that have a higher human relevance, where we can better predict the human situation. And of course, from an ethical point of view, we would like to reduce the number of animals that we're using, with, of course, the ambition or the dream for the future that one day we will not use any animals at all. So this is what really drives us, exploring those newer human cell-based models. Now, what is important, what is interesting for us when we talk about organs on chips? We think organs on chips have the potential to address questions that currently cannot be answered using current models, be it cell line models, other types of experimental systems. What is nice about organs on chips is that typically you can have different types of cells that make up an organ within one system. You have a three-dimensional architecture, which is very important if you think of certain organ types like blood vessels, the gut, and other systems where the 3D architecture is really, really important. There's certain organ chip systems where it's possible to incorporate flow. Flow can be very important if you want to mimic blood, for example, but also, as you can see here in the sketch from the Wyss Institute, air can be important if you're aiming to recapitulate a lung, for example. And maybe a bit more for the future, what is also interesting and relevant is you can start connecting these different organs so that you have maybe your little heart, your little lung, your little brain, and you can really recapitulate aspects of human physiology. So these are the key aspects, together then with other things which are more from a biology point of view, which are relevant. So we have also the extracellular matrix. We can allow cell-cell interactions. We have the immune compartment in there, which especially for us at Roche, we believe is really important, and I will allude to that later on. We can culture those systems for a long time, so not only like 24 hours or maybe 2 days or 3 days. We can really do long-term experiments. We can use such systems to, for example, test drug combinations. As you certainly know, there's, for example, in oncology, very often drugs are not given as a single drug, but you typically have combinations. This is something that is inherently difficult if you want to use animals for that. Of course, also biomarkers is an interesting aspect. You every now and then want to test for your efficacy, but also for the safety during clinical trials, and to develop biomarkers using such human tissues can be of high relevance. Now, when we look at the drug development world today, so typically those in vitro systems are used to prepare for preclinical animal tests. Yeah? So in earlier times, as I showed you in the introductory slide, cell lines have been used, and now many pharma companies are using these type of modern systems, 3D systems, organs on chips, microphysiological systems, to better characterize the candidates before you go into the normal preclinical animal testing and then preparing for entry into human. Now, there's nothing wrong with that, but actually we believe if we really want to make a big step forward, if you really want to change something fundamentally about drug development, we should actually make a much bigger step forward and think broadly, how can we best apply those microphysiological systems? And so the vision we have for the future is that our starting point should always be the patient. Yeah. So be it healthy people or also patients. So we want to start there with exploiting the data that is available, so real-world data from patients, and also getting tissue samples from those patients. And in a so-called reverse translation approach, we would like to use these two elements to establish and also validate in vitro models, human relevant cell models, organs on chips, organoids, etc. Once we have confidence in those models, we would like to, in an ideal world, we would like to directly use these types of systems to predict the human situation. So rather than just introducing organs on chips as part of the normal existing drug development paradigm, we would actually like to change the drug development paradigm. And this is our ambition here. So where do we see application of those models during drug development? So certainly in the early phases, if you imagine you have a model that is constructed using human tissue directly coming from a patient, we believe those models are excellently positioned to study disease-relevant human biology, the pharmacological mode of action of specific drugs, and also, of course, if you have access to diseased patient material, that you use such a model to identify novel drug targets. We also think that rather than using then long screens and also animal testing, we think for the phase of identifying the right lead and also optimize one, we like the concept of the in vitro therapeutic index, so that we test using such human cell-based models, both for their efficacy, but also for their toxicity, and make there the selection directly. So rather than sequentially, as it is today, where you first select a candidate based on its efficacy, and once you have a highly efficacious candidate, you start characterizing it for potential side effects. Of course, later on during preclinical development, where there still will be animals needed, at least to some extent, we think those models can support those animal tests to address key questions for both toxicity, if there's something happening in an animal test, is it human relevant, and also to establish a solid PK/PD. This can be relevant for normal drug development programs, but if you think, for example, for pediatric drug development where you cannot easily test that in animals, we think these type of models can be of real help. Once you are in clinical development, before you certainly can support enabling entry into human, We think combination therapies are an excellent area where these models can help. And also, if you think of establishing models that really reflect an individual patient, we think for personalized healthcare, in our case, personalizing safety with individual models representing an individual patients, I think these models can be really, really useful. Now, if we come to Roche, what is really driving applications of organs on chips and microphysiological systems in our case? So what we have observed is a shift in our portfolio away from classic small molecules to way more complex engineered large molecules that often have multiple targets. And as you can see on the right-hand side, the top graph, so these are the types of large molecules we're dealing with in industry nowadays. So these molecules are quite different from conventional antibodies that all of you know when antibody therapies were established in the 80s and the early 90s. So these are very specialized molecules. They very often do not cross-react with any preclinical species, not even cynomolgus monkeys, which are typically the species of choice for these type of programs. Furthermore, the targets and the pathways which we are typically interested in very often have an immune aspect. And everybody that is involved in immune biology knows that mice, for example, quite much differ from the human situation as indicated in the middle sketch here on the right. So mouse and human biology, especially for immunology, is very much different. So we would have difficulties in using a mouse model for such type of questions. And there's a certain range of general challenges that we are facing. For example, there is a need to assess both the safety and also the efficacy of our drug candidates for children. How should we do that? We don't want to give those drugs to children without knowing, is it safe for children? How would we do that? There's certain ways to do that in juvenile animals, but it's very difficult to make a solid prediction. So could those systems be of help? There's also a growing need to assess the safety and efficacy in different ethnicities. Many of the clinical trials that the big pharma companies are conducting, they include Caucasians in their trial, which is certainly a good starting point. But what does this tell us about the safety and the efficacy in other ethnicities? So we should really start thinking about how to do that. And again, could those organ chip models be of help there? If you think of the drug development world, which more and more also tries to address rare diseases which are not affecting a large population, could those models help in filling gaps where you don't have enough patients? Can you enrich your database by producing organ chip models that represent this patient population? And generally, if we want to increase the benefit risk for patients, medicine should become more personalized, and this is what I'm showing in the bottom right graph. So instead of just one size that fits all, modern drugs will be very tailored so that you have a very specific treatment for individual patients or populations. And we believe these organ chip models can be an excellent tool to test for these aspects before going into clinical trials. So overall, I hope this convinces you that the existing conventional preclinical in vivo testing models may not be relevant, or in some cases simply are not possible. So there is an urgent need for novel tools to assess the pharmacology and also the toxicology of those new types of drug candidates. And I'm showing you now a couple of examples. So we are working a lot with T-cell bispecifics in oncology. So for those who are not familiar with these types of drugs, so these are engineered large molecules which at the same time can target the cancer cell and the T-cell. So the idea is that the large molecule binds to a T-cell, activates it, and brings it to the site of the tumor where it induces killing. And this can have three types of effects. The first one being you have a so-called on-target, on-tumor cell killing. So this is, of course, the desired effect. You would like your drug to go to the tumor and kill the tumor. Unfortunately, the world is never perfect, so sometimes you also have on-target killing, but not on the site of the tumor, but in a healthy part of the human body where the drug target, unfortunately, is also expressed. How do you address that? And should your molecule not be super specific for that particular target, you may also be facing off-target effects. So we are asking ourselves, can we build models that can help us recapitulate such a complex process where a bispecific antibody is recruiting a T cell and brings it to the site of action to induce killing? Can we use such models to assess how do our molecules work and are they safe and efficacious? So over the last years, our teams have started to develop models that recapitulate the wide range of toxicities immune-engaging antibodies can provoke. So in the very early days, it was believed that large molecules are so specifically designed they wouldn't exert any toxicities. But we have learned over the past decade of immune therapies that actually this is not the case, that we do see actually with many of those well-known mainly checkpoint inhibitors, but also other types of T-cell engagers. We see toxicities in a wide range of organs. So we said we need to be able to test that in vitro in human systems and started to build immune-competent models in our labs. How would we apply those models? So these are among the first data we started to generate in early days. So this is a real case example from preclinical development. T-cell bispecific antibody was tested in a cynomolgus monkey study. It was known that actually the target is expressed in the lung, as you can see here in this sketch, but it was believed, well, actually, this molecule shouldn't be able to cross the epithelial barrier of the lung and enter there to cause toxicities. But unfortunately, the team had to learn in a real in vivo experiment, yes, it actually can. So this molecule was terminated, but the team, of course, said, well, for the follow-up molecules, we don't want to use the monkey as our screening models. Is there a way to recapitulate this lung situation here on an in vitro level so that we can use that to test the follow-up molecules? So the in vitro approach designed was we need a perfused vascular system and a lung system which are connected on a membrane. And this is what you can see here on the lower right-hand side. So this is the organ chip model that we have been working on together with Emulate, one of those startup companies that are leading the field of organs on chips. And it consists of pulmonary endothelial cells together with PBMCs recapitulating the vasculature. Then you have a membrane, and on the other side of the membrane, you have the alveolar epithelial cells. So this is our system we use to recapitulate a human lung. And so we started to apply the system. This is actually a picture from the top lung compartment side. And we used that antibody that caused the toxicity. And we were actually quite happy to see that with increasing concentration of the T-cell bispecific antibody, we started to see more and more dead lung cells. So indicating that indeed this complex mechanism of the T-cell bispecific activating a T-cell migrating through the membrane and there hitting the target and causing toxicity, we could recapitulate that. So that was the very first but important step to see using such models, we can start investigating highly complex biology and use an in vitro system to optimize our molecules so that once we move them into later development, that we have a good confidence that they are safe. Moving to another example. So this is actually a well-known example from the literature. It's about a T-cell bispecific called urelumab. It's targeting CD137. And in December 2008, a clinical trial had to be stopped because of hepatotoxicity of two patients, actually fatal cases. And that was came again quite as a surprise, because it was the target CD137 was known not to be expressed in liver. So people were wondering why do we see now toxicity in the liver? It's expressed on activated but not on resting T cells. So what happened here? What you can see on the left-hand side is a study that has been published in the literature where a mouse surrogate was used. So we thought, okay, can we recreate a situation where we have both the target organ, which is the tumor, and the off-target organ, which is the liver, together in one system and see how much tumor killing do we get and how much liver toxicity do we get, and can we use such a system to optimize such a molecule like urelumab. This is the system that we have built up together with the university, with the technical school ETH. That was part of a PhD thesis. You can see on the left-hand side, this is the basic concept. It looks a little bit like a bone. You have this joint compartment where there's both the liver and the tumor represented in there and circulating blood cells. You can see on the right-hand side on this movie here, so this is one part of the system where you have this round spheroid structure, so this is the liver, and you have circulating immune cells here. So that required quite a bit of engineering work, but once we had a prototype ready, we said, okay, can we now recapitulate the toxicity of urelumab? And again, actually, as you can see here on these pictures, so this is the liver compartment we're showing here. We were quite happy to see that actually with increasing amounts of this bispecific antibody, we would also see increasing dead hepatocytes. This is on the top panel from left to the right, from vehicle to increasing concentrations. What was even more important to see is actually that with increasing concentrations of that bispecific, we saw immune cell infiltration. And that seemed to be actually the underlying cause of urelumab's toxicity, that urelumab would bind to T cells, they would get activated, and they would migrate into the liver and there cause toxicity. So again, I'm confirming that we can recapitulate such a complex in vivo situation using a human-relevant in vitro system. Another example I would like to show you here is a bispecific antibody-induced gut toxicities. This is a real case from our portfolio. The traditional system that one would use would be a transwell co-culture of intestinal cells and PBMCs. We couldn't really see here strong toxicity, even though in the clinic it was severe and dose-limiting gut toxicity. We saw only minimal T-cell activation and no damage to the epithelial compartment in an in vitro system. So we thought, okay, can we have an improved in vitro system that is able to recapitulate this toxicity? So we started to generate organoids that we would get directly from patients, and I would first check, as you can see on the top picture, whether the target is expressed, and we would start co-culturing then with PBMCs in hydrogel microwells. And as you can see then, after applying different versions of the drug to this system, so the original drug that was in the clinic, but also a version that didn't make it into the clinic, which has a weaker binder, and a control antibody. And as you can see, we see with the original molecule that caused the toxicity, we see, as outlined here on the left-hand graph, we saw an increased release of interferon gamma, so cytokine release, which was also observed in the clinic, and also with increasing concentration, increased cell death. So it was really nice to see that the model recapitulates the antibody-induced toxicity at clinically relevant concentrations, and it also could differentiate different versions of the molecule. So it was not a yes-no answer or something that would be causing toxicity no matter what molecule you're using, but it was really important to see it's specific, and if we're using a weak binder, like in this case, we would see less effects. We also could confirm that actually the toxicity is intestinal region specific. So it was something that we could confirm by IHC, so that the target is highly expressed in the colon, but only at very low levels further down in the small intestine. So we prepared organoids originating both from small intestinal regions, but also from the colon. And we compared the same specific bispecific antibody, and as you can see in the graph here on the lower left, again, we could recapitulate regional specific toxicity. Again, opening up, of course, ways to mitigate such toxicities if you have a better idea where will it happen and you know that it recapitulates a clinical finding. If we continue with the organoids, I would like to give you a bit of an outlook. Where are we currently working on? What do we have in mind? So organoids are a very powerful model. They can be generated from crypt resections from patients. They can be passaged, they can be expanded, and they can be banked. So that opens up the possibility that you can start building up a donor bank, and you can always take the cells out and use them to establish, for example, an organ chip model. And what we would like to confirm now is that we have a characterized donor. We have the patient history in the background. So do we also see, using those different types of donors, do we see the same diversity of drug response as we could see in the clinic when those different patients received a drug? So this is really a way to address more personalized approach towards drug development by using such donor-specific models that can be expanded and passaged and characterized. So this is just a movie on how we do organoid-based toxicity screening. So in summary, we have in the meantime established a whole suite of human models. They all are built based on primary human tissue. They're polarized where required, and we have the PBMCs from the same patients in a flow-based system. So we have typically the ability for imaging over longer time periods and also media analysis. We think those types of models can be used for both efficacy and for safety profiling. Sometimes we have both the disease element as also the healthy tissue in the same organ. We can use that for antibody format selection. I alluded to the personalized safety approaches. We also think that it can support clinical trial design. If you think of getting tissue samples from patients you're going to treat, and you can also optimize the choice of your combination therapy. Of course, as explained in the very beginning of my talk, you can also use such a patient-specific model for discovery and target evaluation. Here's one last example what we're currently working on to meet our ambition of animal-free drug development. So here's another bispecific, which is not cross-reactive to any animal species. We know from our genomic database that not only is the target expressed in the tumor tissue, but also in healthy tissue, in this case, in podocytes of the kidney. Yeah. So in order to establish a good safety profile of this molecule, so what we first did is just compare, of course, the tumor tissue in vitro, but also the known podocyte tissue. This is the graph we received. And as you can see, because the target is much more highly expressed on the tumor as compared to normal healthy cells in the kidney, killing is much more efficient there. But of course, this is not sufficient for a real safety profiling. What about all the other organs? So we said we're using now a suite of just the key organs of the human body and compare to our tumor killing to generate an in vitro therapeutic index. So we took organ systems representing the brain, vasculature, heart, lung, liver, intestine, and kidney, together with PBMCs, and started to profile the molecule. So without going into details, as you can see, we had then a whole suite of established killing curves in the different organs that really help to characterize such a molecule. And currently, this molecule is about to enter into human, supporting a so-called MABEL approach. So the minimal anticipated biological effect level, where we are in discussions also with authorities, whether such a safety profiling characterization in the absence of any animal test can really help moving such a molecule into patients. So what are we currently working on? We're continuing to generate immune competent autologous models where we have tissue and blood from the same donor. We want to include both the healthy and the diseased tissue and want to do combining disease pharmacology and safety in vitro, as I showed you in the example before. We want to use those models to support personalized approaches, be it individuals, but also patient subpopulations. Can we identify subpopulations that might be at risk? Can we address ethnic differences? Can we use such models for juvenile versus adult testing? We would like to automate, of course, those models so that we have an increased robustness, but also throughput. And we want to improve the access to human tissue. This is still, of course, difficult to get directly out of clinical trials or from hospitals, patient-derived tissue. That is not easy, and we need to work on that jointly. So with that, I would like to sum up and show you this is basically our approach. So we're not only using these complex systems. It really depends on the questions. For some questions, a cell line might be the absolutely right thing to do. If you know exactly that your cell line has the target expressed, it has the biology you need, there's no need to go for any more complex model. It really depends on what is your question, and this defines which model you are using. So this is a bit our mantra, that it's the biological question that drives the selection of the cellular model and the readout. So it's always following the rule as simple as possible, but as complex as needed. So for very complex questions like in cancer immunotherapy, we feel it is necessary to have more complex models, and this is what we're following. There's a couple of pros and cons around those organ chip models. So I talked a lot about the opportunities like combining pharmacology and safety, internal decision making, where we already apply those models. I think it's important that we should always balance the application, that we really aim for reducing animal testing and not just add more tests and still do the normal animal testing. Yeah. We think it can support entry into human and also clinical trials and personalized safety. Now, the current challenges are, of course, These models are very complex, and the demonstration of the physiological relevance will be increasingly difficult with increasing complexity. It's not always easy to really demonstrate once you have established such a model, is it relevant? How do you show that? And this increased complexity also comes with a price. Yeah. It's a technical complexity for the people in the lab to deal with those kinds of models. They have a lot of variability. Sometimes they are not so robust. So this is something to be aware of. I alluded to the sourcing of human cells mainly, sometimes also animal cells. This is very important, and this can be very challenging if different cell types from the same human donor are needed. Of course, like for any other in vitro system, the translation from in vitro to human is key and is not so easy. And most of these models are only semi-validated. They are improving and improving, but this is not so easy just to demonstrate that actually you can translate to a real clinical human situation. A lot of work has been done, good work, and we are on track, but a lot of more work needs to be done still. With that, I would like to also show you a quote I like a lot, because there's a lot of discussions. Well, are these models already perfect? Is it really exactly like a human organ? No, of course not. These models are imperfect. But as Bob Weinberg said, just because they're imperfect, it doesn't mean they are wrong. We just need to be aware. Where are the challenges? Where are the opportunities? How well is it established? And where are the shortcomings of such a model? With that, I would like to close and thank people from my former department, also people working with me now in my current department that helped building up these models, and also some of the external collaborators which helped us really doing pioneering work. And I hope that was informative and instructive and interesting for you. We cannot have a Q&A right now, but I'm also happy to take any questions later on, be it by mail or via other sources. So thank you very much for your attention. And with that, I stop here. Thank you.